In this module, we look at Mendel's experiment. We will see what was Mendel's question, how he designed his experiment, and how he was able to conclude what the conclusions of his experiments. So before we get to that, let me introduce to you the Mendel's subject. Mendel was working on pea plants. These are the garden pea plants. These plants are easy to cultivate. They have short lifespan and they have several characteristics and several traits that Mendel could follow. Flowers are the reproductive organs of these plants. As you know that most flowers have both male and female reproductive organs. The ovary is the female sex organ. You can see it on your screen. The stigma is the part of the flower where the pollen lands and extends the tube. Pollen is sperm equivalent of the plants. Anthers are the male sex organs. These are attached to stamens, which are filamentous structures, and anthers produce pollens which can then land on stigma and fertilize the egg inside the ovary. So Mendel used these plants. It was easy to grow them, as I mentioned. These plants can also be controlled in such a way that if Mendel wanted, he could remove the male or the female sex organs, or he could self-pollinate the plant itself. The technique Mendel used was simple. He used a paintbrush. He rubbed it on the anthers and he collected pollens in the tip of the brush, and later he dusted on the stigma of the female part of the flower. As a result, the pollens fertilized the egg and resulted in seeds which he could grow and look at the next generation of the cross, of the breeding experiment between two types of flowers. The first experiment he did involved the shape of the seed. Mendel noticed that there were two types of seeds, a spherical shape and a wrinkled shape. Before he began his experiment, he wanted to ensure that the plants were true breeding. What do I mean by that? He crossed a typical plant with a typical trait, a specific trait, with a plant of the same trait, with the same specific trait. So, for example, he crossed plants with spherical seeds with other plants with spherical seeds for several generations to ensure that now the plant is true breeding. The trait it carries is now completely spherical. He did the same exercise with the plants that had wrinkled seed to ensure that these were also true breeding plants which produced wrinkled seeds. Then he cross-pollinated a plant that was going to produce spherical seed with a plant that was going to produce wrinkled seed. So these plants were the parental plants which were true breeding, and they're donated parental plants, or P, abbreviated P. Mendel got the first generation. He got the result of his experiment produced all spherical seeds. Now, if the blending concept was correct, all these seeds would be semi-wrinkled and semi-spherical. It was as the wrinkled seed trait had completely disappeared, because none of these plants of F1 generation, which stands for first filial generation, they had any wrinkled seeds. So here we could see the blending theory was incorrect, because blending theory would have predicted that the offspring would have mixture of characteristics from both the parents. Next, he crossed the F1 generation among themselves, and he got the F2 or second filial generation. He saw that in the F2 generation, the seeds, 3 out of 4 seeds were spherical and 1 out of 4 seeds were wrinkled. So what were the conclusions? How he used this data to interpret the results and how he was able to make specific conclusions about the particulate theory. We will look at that in the following segment.